Blanco lab, we study a disease called autosomal dominant polycystic kidney disease, or ADPKD. Patients with this disease have many cysts on their kidneys, and these cysts grow larger throughout the patient's lifetime. Eventually, they get so large that the kidney function is compromised, necessitating dialysis or kidney transplant for our patients. Studies show that about 1 in 1,000 people have this disease worldwide, making it the most common inherited kidney disorder, and also the fourth leading cause of end-stage renal disease in the United States, or kidney failure in the United States. Despite its prevalence, there's no cure for this disease and very limited treatment options for our patients. What makes this disease challenging to study is that patients with the same genetic mutation or the same thing causing the disease can have vastly different outcomes. For example, one patient may have preserved kidney functions into their 70s, while another of the same family might have kidney failure decades earlier. And this means that things besides genetics influence the outcome of the disease, such as environmental factors. In the Blanco lab, we study these environmental factors, or things circulating in the blood, that influence the progression of ADPKD. Specifically, we study a hormone called wabane. Wabane is produced in the body as a hormone, and it's also consumed from the diet from plants. We've found that wabane will increase the cyst growth and size inside kidney cells. And that's specific to ADPKD kidneys, so it's a malfunction of how ADPKD patient kidneys are functioning. For example, if you take ADPKD patient cells and you apply wabane, you see an increase in the growth of cells, as well as the fluid secretion, two characteristic hallmarks of ADPKD cysts. When you do the same thing to normal human kidney cells, you don't see any changes in cell growth or fluid secretion. Recently, I've taken these studies to mouse models to validate that we see them in a whole organism. In ADPKD mice, wabane will increase the number and the size of cysts while having no effect on normal mice. In our lab, we want to discover compounds which can block wabane's actions. We hypothesized that by stopping wabane acting in ADPKD patients, we can reduce the size of cyst growth, or the amount of cyst growth, preserving the lifespan of the kidney. So our goal is to create drugs that patients could take that would act on kidney cells to block wabane's actions, therefore extending the lifespan of the patient and the health of the patient and quality of life. Thank you, and happy World Kidney Day.